Using ancestral protein recreation, I will be looking into the evolutionary past of PETase to try and identify an ancestral protein sequence which may result in a more thermostable protein. By finding this ancestral protein, alternative starting point for the bioengineering of PETase can be used, which will not require any further modification in regards to thermostability. Polyethylene terephthalate is one of the most prolific plastics used currently, with estimates of around 70 million tons of PET being manufactured annually. PET is most notably chemically inert and has great longevity, which has often led to its prominence in both industry and within the commercial household. However, these same properties make it incredibly difficult to process and recycle PET, costs of which are often extravagant and often leads to products with inferior mechanical properties. This has resulted in an increase in plastic wastes often entering our oceans, with current projections estimating at about 33 billion tons of plastics will be produced as waste by 2050. In 2016, a new bacterial strain was discovered in a recycling plant in Japan called Adionella sakaiensis, which was capable of partially breaking down PET to use as its carbon source via secretion of a protein called PETase. PETase was capable of hydrolyzing PET to its more innocuous constituents, terephthalic acid and ethylene glycol, with wild-type PETase being capable of almost fully degrading PET film over a course of 6 weeks. However, the glass transition temperature required for PET is well above the temperatures in which PETase can remain stable in. One method to achieve this thermal stability is through ancestral protein reconstruction, which is the inference of ancestral protein sequences which are derived from the bioinformatics and phylogenetic analysis of their modern counterparts. Due to the harsher environment from which these ancestral proteins were theorized to exist in, the ancestral protein is expected to function at a higher temperature and could possibly be more promiscuous, allowing it to possibly hydrolyze more plastics, including PET. To identify the potential ancestral protein sequence of PETase, it is imperative that a phylogenetic tree be generated from existent homologous protein sequences. To find this sequence, I used an amino acid sequence of a PETase mutant called chain A polyethylene terephthalate hydrolase, or 5XJHA. Using the T-BLAST-N function from the NCBI site, which uses a protein sequence and translates it into a nucleotide sequence, a search inquiry with 5XJHA as the subject was inquired using a nucleotide collection database with a max target sequence of 500. To help filter the search results, set the percent identity to 45 to 100%, and then the query range to 75 to 100% to decrease the total amount of. Select coding sequences with a small e value is of great importance, as the smaller e value indicates that the alignment was not by chance. To help increase the variety of sequences chosen, limit selections to 4 species per genus. Having selected your homologous protein sequence, download the FASTA aligned sequence from BLAST to use for MEGA. After having converted the .txt file into a MEGA format, create a new alignment session using the converted .txt. Then use muscle to align by codons to ensure that the coding sequence remains functional. Using the aligned coding sequences, create a maximum likelihood phylogenetic tree and extract the ancestral states and show node IDs. Using the additional program from phylogenetic trees made easy 5th edition student resources, which then separates the original ancestral states file into separate instances per node and helps determine the most likely nucleotide base and protein sequence. As can be observed from the phylogenetic tree, nodes 55, 57, 68, 70, 72, 73, and 79 are all nodes from which thermophilic bacteria, Thermomonospora curvata and Thermobifida siliciotic all evolved from, with nodes 55 being the most common ancestor for all bacteria chosen. Using the aligned homologous DNA sequences from the 41 different coding sequences that we got from NCBI, a rooted maximum likelihood phylogenetic tree was generated, which deduces the phylogenetic tree based on generating a sequence of the likelihood of a base to occupy any given space, the probability of which was made in reference to the extant sequence provided from BLAST. Vertical analysis of the phylogenetic tree are in respect to Thermomonacurvata and Thermobifidia silicita shows that nodes 45, 55, 57, 68, 70, 72, 73, and 79 are of interest, as these nodes all contain crucial but subtle amino differences which may result in a more thermostable protein. be noted, however, that the accuracy score is not indicative of the probability of this sequence to actually be the real ancestral protein, but is an average of the probability of the most probable base. It should also be noted that the actual probability of the both ancestral protein sequence and protein itself would decrease the further back we look into the evolutionary history. DNA sequence at node 55, for example, is at 7.70 multiplied by 10 to the negative 56, whilst its most recent ancestor between Idonella and Acidophorax is 0.24699. In conclusion, tracking back the evolutionary history of Thermomonospora curvata and Thermobifidia celestica, both of which are thermophilic and related to adenylcyanosis, that the ancestral protein sequences derived from the maximum likelihood phylogenetic tree nodes 45, 55, 57, 68, 70, 72, 73, and 79 all may result in a potentially more thermostable protein. To further validate the theory behind ancestral protein reconstruction, recreating these protein sequences and having them being expressed by existing organisms would be of great interest. Testing whether these ancestral proteins are more promiscuous and possess greater thermostability will be of interest, as well as these protein sequences can then be further modified to help increase their efficiency for industrial use.